This is AP Biology, Chapter 27. It's the reproductive system. This is our fourth and final video. Previously, we have talked about sexual and asexual reproduction. We've talked about the anatomy of male and female reproductive systems. We've talked about the menstruation cycle and the hormonal control of the reproductive system. And this is our fourth and final video. We will be discussing fertilization and the development of the embryo. So, fertilization is what kind of results in the zygote, and the zygote then triggers or develops through embryonic development into our full-fledged baby. When we have a sperm joining with an egg, it's interesting to think that there's only one sperm that's able to get into the egg, when in fact there's thousands of sperm that are released in an ejaculation, any of which can possibly reach the egg. This is a microscope slide actually showing how many sperm are attached to the exterior of one egg, trying to be the one to fertilize. When we look at fertilization and how that process occurs, because the egg has a cell membrane on it, what happens is the sperm are designed not only for mobility to swim and reach the egg, but secondly, they're designed to actually pierce into the egg and reach the egg's nucleus. So we can see that this sperm has a cell membrane that is elongated into a tail, which is a flagella used for swimming. It has a lot of mitochondria, which provides energy needed for movement to swim to the egg. There's the short neck region, and then inside the head region, you'll find the nucleus. This is where the haploid DNA is found. There is also something called the acrosome. Now, the acrosome is actually what is full of enzymes that can digest and break through the egg's protective coating, allowing fertilization to occur. So once sperm surface proteins, so proteins that are found on the cell membrane of sperm, bind to the egg receptor protein, they will fuse together, igniting the two cells to merge, allowing the nuclei to find each other and fertilization to occur. What happens is that as soon as this binding between sperm and egg receptors on the membranes occurs, the egg property kind of changes, and it prevents any other sperm from possibly getting into that egg. The one sperm that did join and fertilize then allows the zygote or the embryo to develop. So here is the process of fertilization. You can see here's our acrosome full of digestive enzymes. The receptors on the membrane and this sperm bind together. The acrosome enzymes are released, which then allows the digestion to occur. So now the receptor membranes are fully activated. We are now releasing the nucleus into our egg. The egg nucleus joins with the sperm nucleus, resulting in our zygote, which then develops into our embryo. So here's our process. Now, once we have a zygote, what happens is it rapidly starts going through cell divisions of mitosis, creating or ultimately leading to a solid embryo. So one cell quickly becomes 2, 2 become 4, 4 become 8, 8 become 16, and so on. Eventually, we go through enough cell divisions to create this solid ball. That solid ball then hollows out much like a soccer ball would, and it becomes known as a blastula. The blastula is important because it's where we actually start orienting the development of an embryo. Otherwise, we would have heads coming out of eyeballs and things like that. Once we have our blastula, a process called gastrulation happens. And gastrulation is where cells start moving to start forming the 3 different tissue layers of an embryo. One of the major things is let's start developing the digestive cavity that runs the entire length of an organism. Once we have gastrulation, we're going to get 3 distinct layers of cells, and each layer can only become certain parts of a body. So here is when we look at a frog gastrulation. So the first thing that's happening is our blastula. We're going to go through gastrulation, so cells are going to start moving, things are going to start orienting themselves. Through gastrulation, what happens is we start to form all of the different tissues or layer cells. Once we've completed gastrulation, we can clearly see ectoderm, endoderm, and mesoderm. With that, we now have orientation within the animal. Now, after gastrulation, all the organs start to show up. So one of the major ones is the backbone or the nodal cord. And so you can actually see in this diagram right here, the nodal cord starts to develop here with the neural fold and neural plate. Now, what happens is these cells start to differentiate and move, thereby allowing an actual hollow neural tube to happen, and this neural tube later becomes the spinal cord. So here we are. This is our ectoderm. The cells move and pinch in, creating this hollow pipe cavity with the ectoderm on the outside of it. That ectoderm becomes our skin, and the neural tube becomes our backbone or spinal cord. Once we've done that, then we start making our coelom, and our coelom is what becomes our internal organ body cavity. We also form the archerenteron, which is part of our digestive cavity. So now we're starting to make the digestive system. We'll make room for the respiratory and the nervous and all of that. If we look at a frog is a really cool thing because during one stage of development you get a tadpole. From a tadpole you get a polywog. From a polywog you eventually get your adult. All of those changes have to occur in order for the life stages to occur for a frog. If we were to look at what are those embryonic tissue layers, the endoderm, ectoderm, and mesoderm, what do they turn into? Well, the ectoderm mainly becomes the skin or the lining on the outside of things. Your endoderm is becoming the lining on the inside. So this is like the epithelial lining that's in your small intestine. That's part of your digestive tract. It's the inside of your lungs, your respiratory system. It becomes your liver, your pancreas, your thyroid, so all those endocrine glands. It becomes the inside of your bladder, which is for your excretory system. The mesoderm, on the other hand, becomes your skeletal system, your muscular system, your circulatory system, and then, of course, the nodal cord, which is the spinal cord. Now, if we were to look at this, how does this happen? Well, we have to have cells change shape, they have to move, and some of them have to die in order for proper cell development to occur. If it doesn't happen, if this proper cell death doesn't occur, what happens then is we get defective cells. And the cell death is called apoptosis. Apoptosis is critical for proper development. So what happens is you have some cells, a cell will die because it's programmed to do so, and then it gets engulfed and digested by another cell, which then allows the proper cell to grow and develop. Now, one of the big things that happens with this idea of how do we make organs from just tissues is something called induction. And what happens is one cell tells the cells next to it, hey, I'm going to become a heart cell, you should become a heart cell too and start pumping with me. How do they communicate? Through cell communication with reception, signal transduction, and response. So here is the example of like your future brain and eye. So what happens is some of the cells to say, hey, I'm going to become an eye. The cells nearby start to develop correctly. All the cells move, migrate, apoptosis happens, and then you get the proper development of all the parts of an eye structure. So this is how we start to form organs and kind of get the parts that we need for organ systems. So it turns out that the parts that become organs in an animal follow a kind of a correct structure pattern, and that's where orientation comes from. So they know that if you were to look at an animal's, and you'll see this little bump right here in between the ventral and proximal areas, you'll see the limb bud. The limb bud, these cells will become all of the parts necessary to make a limb, for example, all the parts that make up a wing. If you were to take the limb bud and move those cells to the anterior portion, guess what would form at the anterior portion of this animal? It would become a wing off the top of the head of an animal. Now, experimental evidence has been done to test this, and here's what they have found, is that there are zones of cells that tell other cells, hey, here's what we're doing. So here's an example. They've taken a donor, a bunch of limb donor cells, and they've grafted into a new area of a developing embryo, and guess what happens? You get two wings because you're telling these cells think they have to become a wing. They don't know what position they are in relative to the other cells. Now, what happens is that in the development of a human embryo, we're talking about pregnancy. Pregnancy is sometimes called gestation, and basically it's where a female reproductive system develops and nurtures an embryo until it's of a stage that it can then survive on its own. And so here we looked at the process of ovulation. So the spike in follicle stimulating luteinizing hormones releases the oocyte. It travels through our oviduct, which is the fallopian tubes. It travels through. It gets fertilized by a sperm. We now have our zygote. It starts to go through mitosis and develops. Once it implants itself into the endometrial lining of a uterus, then we are at the blastocyst stage, which then can start to develop those tissue layers. The tissue layers then happen where we develop the placenta and we have our embryo developing. Now, the blastocyst outside most part, so those outside area that you can see on a blastocyst, the hollow ball, is called the trophoblast. And the trophoblast is actually how an embryo implants itself or attaches to the uterine wall. Once it's there, what else is happening is that there's these other membranes that are starting to develop, and they're called the amnion, the carrion, yolk sac, and the allantois. And what happens is that they're all part of the necessary process for nurturing that embryo. Now, what do they do? Well, once they're fully formed, it's how we turn into or develop the placenta. It's how we make our umbilical cord. It's how the female reproductive system can nurture the growing and developing embryo. Now, the embryo is actually in itself floating in this fluid-filled amniotic cavity, which is surrounded by the amnion. And what happens is it allows that embryo to reach and absorb food and oxygen from the mother's blood. Now, one of the big things is that the gestation period for humans is developed or divided into 3 different trimesters, so trimester 1, 2, and 3. First trimester is where there's rapid changes that are occurring. We have a lot of development that occurs. The limbas, the brain, the eyes, the blood system all start to develop. By 9 weeks, we no longer call the embryo an embryo, we call it a fetus. Once we're in the second trimester, which is about 3 months in, we're now just going to be getting bigger in size, and we're going to start refining or kind of developing further those features of a human fetus. This is where it looks less like an alien and more like a baby. At about 20 weeks, this is when usually they do ultrasounds or they take pictures of the baby. Usually it is about 19 centimeters long. It weighs only about the size of an almond, but you can start to see it has reproductive structures. By the third trimester, this is when the baby just gets bigger and fatter and then ultimately comes out through birth. Childbirth is one of those hormonally controlled things, and it actually is brought on through muscle contractions of the uterus, and that's what we call labor. One of the major hormones that is used to allow for labor to occur is estrogen. So the ovaries release estrogen, which then induces oxytocin in the uterus. What happens is oxytocin is used to stimulate the uterus to contract, so it actually causes the uterus to squeeze. What also happens is it causes the placenta to make something called prostaglandins, and what that means is that actually causes the uterine to contract even more. So the hormones are causing even more hormones that cause even more contractions. This is a positive feedback mechanism, and it continues until ultimately the baby's out and the uterus no longer has a need to contract. So here's the first stage. There's dilation, expulsion, and then ultimately delivery of the placenta. So during dilation, this is when the cervix is expanding. During delivery, this is where the baby actually comes out away from the uterus. And then lastly is where the placenta, so that lining of the uterus, has to be released from the female so that then her body can recover. Now, one of the things is that if you're unable to have babies, that's okay, because we now have fertility treatments that can significantly help and provide infertile couples with offspring. And some of them have ethical and legal questions, but for the most part, it's widely accepted at this point in time. That's it.